Oh, good evening everyone, and thanks for sticking around. My name's Cameron Fraser and I'm an honours student from the Department of Ophthalmology. I'm here tonight to talk to you about an exciting new technology developed right here at this university. There's a new concept in medicine of regeneration. So instead of treating diseases with drugs or surgery, the idea is you restore function by repairing or replacing damaged cells and tissues. In particular, there has been a lot of research into developing better treatments for common neurological diseases. Some of these include glaucoma, stroke, spinal cord injury, and Parkinson's, which affect hundreds of millions of people worldwide. In fact, there are over 100 clinical trials currently underway looking at regenerative treatments for these diseases. However, most of these new treatments require a source of cells, and all of the current sources of cells have a variety of disadvantages. You may have heard the controversy surrounding embryonic stem cells. However, they don't just have the ethical concerns surrounding them, they also have the problem of tissue rejection. You're taking cells from one individual and transplanting them to another. That's why people developed induced pluripotent stem cells and direct cell reprogramming, which overcomes these advantages. But it brings up new problems because it involves genetic manipulation and is a highly inefficient process. It's also worth mentioning adult stem cells that have their own disadvantages, like being difficult to isolate and limited repair potential. However, a new technology has been developed at the University of Auckland to try and overcome some of these disadvantages. It's been found that keratocytes, a cell from the eye, can be reprogrammed into neurons simply by exposing it to 3 specific growth factors. This has the advantages of not involving any genetic manipulation. It allows you to take cells from a patient and in theory give it back to the same patient. And most excitingly, 100% of the cells start reprogramming within 3 days. However, it has one major disadvantage, and that's quantity. You can't take many cells out of someone's eye without compromising their vision. So that's where I come in. That's my project. Find a way to make more neurons using this technology. For those of you here that aren't ophthalmologists, just to give you some context, the cornea is the transparent structure at the front of the eye where the keratocytes reside. It has 3 layers. The middle layer is called the stroma, and it's jam-packed with keratocytes, the cells that we're reprogramming. So there's a few challenges to making more neurons. Chiefly, keratocytes, they don't proliferate, which makes making more of them quite difficult. So the initial approach to my research was find a way to proliferate these keratocytes. However, every time I got the keratocytes to proliferate, they turned into this other closely related cell type called corneal fibroblasts. So I thought, why don't I let the problem be the solution? Can I find a way to reprogram these proliferative and abundant corneal fibroblasts, which will then solve this issue of quantity? And the results from my research strongly suggest that yes, these corneal fibroblasts can be reprogrammed. So to answer these questions, I used a combination of mammalian cell culture, immunohistochemistry, and I also formed a collaboration with the neurophysiology lab here at the university. I used both human and rat tissue for my research. In brief, I dissected the eyes under a surgical microscope to isolate the stroma where the keratocytes reside. I then released the keratocytes from the tissue with a special cocktail of enzymes, and then I cultured the keratocytes in serum-containing media to turn them into these corneal fibroblasts and to proliferate them so I could make large quantities of them. And then finally, I reprogrammed them. So, what I can tell you from my research is that a highly proliferated population of corneal fibroblasts, when cultured in those 3 specific growth factors listed there, take on a neuronal morphology and start expressing neuronal proteins. After only 5 days of reprogramming, we can see a striking difference in morphology. On the right here, we can see in the control group the classic corneal fibroblast morphology, and over in the experimental group, you can see strikingly different morphology with all these fine dendritic processes, which is what you'd expect from neurons. After 7 days of reprogramming, I start seeing neuronal precursor proteins being expressed, like nestin and neurofilament 200. And after 12 days of reprogramming, I start seeing mature neuronal proteins being expressed, like beta-3 tubulin. However, there's one thing that I haven't been able to ascertain, and that's if these neurons are functional. Neurons are electrically active cells, and this electrical activity can be measured. However, when we've gone to measure this electrical activity, we've found that cells reprogrammed for 7 days don't have the electrical activity you'd expect from neurons. So my thinking is we haven't reprogrammed them for long enough to get that electrical functionality. So the current experiments we're planning is to reprogram the cells for longer and then see if they'll have this electrical activity. So that's the main current direction of my research. And I also want to quantify the proliferation. I want to see, put a number to exactly how many neurons can I make from a fixed, small starting number of keratocytes. Well, there's some exciting implications for this research. I mean, if these reprogrammed fibroblasts are actually turn out to be functional neurons, this will be a novel source of neurons, unlike anything else in the world that has been published to date. Future directions of the technology will be to move on to animal therapeutic trials to see if these cells can be used to restore function in disease. And it's also worth considering what other reprogramming is possible. Recently, one of my colleagues has shown that chondrocytes, cells from cartilage, nothing to do with the eye, can also be reprogrammed into neurons using the same non-genetic method. So it begs the question, what other useful reprogramming is possible with the right growth factors? So in conclusion, a novel source of neurons has been discovered at this university. It has some great advantages like not involving genetic manipulation, being highly efficient with 100% of the cells reprogramming in a short space of time. However, it has the main stumbling block of quantity. However, my research with corneal fibroblasts promises to overcome this issue of quantity. Looks like I have time tonight to thank my supervisors, Colin and Trevor, and also my colleague Carol for all their help this year. I'd also like to thank all the people who donated the Eyes to Science that made this research possible, and also all the sources of funding that also, without this research, wouldn't be possible. Cool. Thank you.